Hello, everyone. Please welcome to my channel, Yashoda Krishna 1392. Today, we are going to learn about anti-tumor antibiotics in continuation to previous video, that is anthracyclines. Among anthracyclines, doxorubicin is the most prominent drug. Apart from that, there are few drugs which are already marketed and approved by authorities, regulatory authorities, doxorubicin, daunorubicin, epirubicin, idarubicin. There are still 25 more compounds in trials, which can come into the market in next couple of years. So now in this important drug is doxorubicin. As we discussed previous slide, doxorubicin is very important drug. Now, this is a glycoside. Actually, these are from the microorganisms, Streptomyces species. Very good antibiotics. Now, these are glycosides. Now, A, B, C, D ring. This A, B, C, D ring. This is aglycone portion. A ring, B ring, C ring, D ring is aglycone portion. And this is the sugar portion, donosamine. This is the sugar portion called donosamine. And both of these sugar and non-sugar portion are linked by together by glycosidic linkage. By glycosidic linkage between 7 and one of the donosamine. Donosamine. Now, so this is glycoside as we learned. See, now this B, C, D rings are aromatic in nature as we learned previous video. Please go through that video in the link provided below. These are aromatic rings, so they can slip between or intercalate between two DNA molecules. DNA molecules. Now, because of this, the anthraquinone chromophore, that is BCD ring. BCD ring is known as anthraquinone. Sorry, anthraquinone group. Anthraquinone group. This is the total. This portion is called as anthraquinone portion, and which is chromophore portion. Chromophore portion. Now these hydroxyl groups, keto group, hydroxyl group, once after entering, because of this chromophore group, once after entering into DNA molecule, they bind to the bases through hydrogen bonding, even these. Now, the sugar portion has an amine group. At the physiological pH, it becomes quaternary. Now, this can bind with ionic bonding with the phosphate group present in DNA molecule. This is how this drug interact with the DNA molecule. So this is the basic structure what we have discussed. Now, so doxorubicin, previously it is also called as andriomycin. It is obtained from from microorganisms from the soil, Streptomyces pusitis in 1967. Now, let us see some of its derivatives which are marketed. This is doxorubicin. Now, you can see at ninth position, we have given numbering. Numbering 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Now, at ninth position, these are the two structures which we placed in this slide, doxorubicin and daunorubicin. Now you can see the structural difference between this doxorubicin and daunorubicin. Daunorubicin, at ninth position, there is acetyl group. There is acetyl group. Whereas doxo, it has oxygen. See, it has hydroxyacetyl group. It has hydroxyacetyl group, which is at the 19th position. That is only difference between doxorubicin and daunorubicin. Doxorubicin is quite active and most potent drug. Potent drug. Now, in next slide, we will see some more drugs or some more its drugs. So here, for our convenience purpose, I placed here again doxorubicin, same structure. Now we are going to learn about epirubicin and idorubicin. These are second generation anthracyclines. These two, epirubicin, idorubicin, are second generation anthracycline derivatives. What is difference here? You see, in epirubicin is similar to that of doxorubicin. If you can see the structures here, it has hydroxy acetate group at ninth position. Same way here also in doxorubicin. What is difference is that the sugar molecule at fourth position, the hydroxyl group is up, whereas here hydroxyl group is below the plane. This is the difference. You people also might have learned in carbohydrate chemistry. What do you mean by epi? Epi means carbohydrates which are differ in one carbon atom. The isomer which differ at one carbon, remaining isomerism at different carbons is same. That's why here you see the isomerism at all the carbons is same to that of doxorubicin except at fourth position. Now here it is below the plane or beta configuration, you can tell beta configuration. Here it is alpha configuration above the plane, alpha configuration. Hydroxyl group is alpha configuration. Or you can also talk in terms of cis trans. Here you see amino group and hydroxyl group are on the same side. That is cis configuration, you can tell. Cis configuration, you can tell. Here amino group and hydroxyl group are opposite, anti-configuration, we can tell. That is only difference between epirubicin and doxorubicin. Now, what is difference between idorubicin and doxorubicin? Now, idorubicin, we can tell by seeing the structure. You can see it here. This is the idorubicin. Check at ninth position. It is having methyl group or acetyl group is present. It is not hydroxyacetyl group at ninth position. That is nothing but it is a derivative of daunorubicin. What is difference? At fourth position, you see at fourth position, there is no methoxy group as usually it is having. That's why we call idorubicin as 4-dimethoxydaunorubicin. Daunorubicin, very good drug in treatment of leukemia. In treatment of leukemia. So these are the structures which are difference between the structures and which are widely prescribed drugs from the class. Now, let us read the SAR of these compounds. So now this is anthraquinone portion, the BCD ring, anthraquinone chromophore, which is responsible for slipping into the DNA molecule and acts as intercalating agent. Now, C4, if you replace, we are telling some important time. There is no any functional group on D ring 1, 2, 3. We are coming directly onto the fourth carbon. C4 modification by demethylation do not affect bioactivity but produce prolonged half-life. One example, as we have seen in previous slide, the structure idorubicin is one of the example which doesn't have which doesn't have methoxy group, which doesn't have methoxy group. Now, transformation of this fifth one, the transformation of fifth carbamyl group to corresponding imine functional group resulted in active compound. Still, there is no marketed preparation, but they're done. I told you in the first slide itself, 25 compounds are in pipeline. So one among that is this one. What is the advantage of these? These drugs has cardiotoxicity effect when there is imine group. There is no cardiotoxicity. So these functional group, especially these two keto groups which are at 5th position and 12th position, are responsible for generation of free radicals. You will learn them in the coming slide. So if you remove them, it is causing less cardiotoxic effect. Now, one more you can see it. It has been shown that methylation at 6 and 11 on the phenolic group, what happens? Reduced activity. These groups, when we discussed about the interaction, these groups are responsible for interacting with our DNA molecule, DNA base pairs by hydrogen bonding. If methyl group, if you add, there is no hydrogen bonding. That is missing. That's why hydroxyl group is important here. Now, ring A bears a carbon side chain. This is the what we are discussing. And chiral hydroxyl group. Now at 7th position, there is glycosidic linkase, which is attached. Now at 13th carbonyl group reduction to alcohol. This is 13. See, after 12, this is 13. If you give a continuous number, this is 14. 14. If you reduce this carbonyl group, you are getting secondary alcohol by the help of enzyme aldoketo reductase enzyme. So if the group is bigger, if this we have kept here CH2OH, if the group is a bigger one, what happens is that you are getting a molecule with long duration of actions. Still, there is no drugs in the market, but this is the what SCR we need to learn. Maybe in future these drugs will come. So what I will explain again, you see, and 13th, I told you what is the 13th. Here you see 12, this is 13, this carbon is 13, this carbon is 14. If this carbon is reduced, if it is reduced, having bigger functional group here, bigger alkyl group, then it is having long duration of action. Then this is about SAR. So what we learned, the basic ring has to be as such, that then the pharmacophore has to be as such. The first one is we discussed about fourth one methoxy group you can remove. Next one, fifth, we told about double bond O that can be done imine group so that cardiotoxicity is reduced. Then we discussed about 6th hydroxyl group and parallel 11th hydroxyl group. When you remove this mask with methoxy group, activity is lost because there is no formation of hydrogen bonding. Then there is chiral carbon at 9th position. 7th position, there is sugar attached. There is sugar attached to this side. Then on the 13th keto group, if you reduce it, having long chain carbon atom, then you are getting prolonged duration of action. This is about the SAR. Next, we will study about the mechanism of action. The main mechanism of action of these drugs is that, see, it has high affinity to bind to DNA by intercalation process. Even previous slide, we told this what it mean by intercalation and all those. Uh, because of intercalation, there is distort DNA shape. So there is a bond breaking between the DNA molecule. And one more is that these drugs block topoisomerase 2. Topoisomerase 2 is an enzyme which helps in unwinding of DNA molecule, or it prevent topoisomerase topological pressure during the process of unwinding. So when there is no unwinding, there is no formation of DNA molecule. Now, one more important mechanism and as well as side effect is generation of free radicals. There is generation of free radicals. So this is our drug. See, we kept here R, irrespective of drugs, and we kept here sugar. We kept here sugar. We didn't put donosomine because epirubicin is different and all those things. That's why we kept sugar. Now here what happens is that the quinone form which was present will be uh, oxidized, to semi-quinone form. Semi-quinone form, we having an electron extra. Reduction form, it is becoming reduction process. Now what happens is that now this semi-quinone form will react with water molecule to form again quinone form. During this process, what happened, the extra electron which was which we got, extra electron which we got here will be transferred onto the oxygen. So that is called as superoxide. Now this superoxide will react with the superoxide is very dangerous, very dangerous. So in the body, there is one enzyme called as superoxide dimutase. Superoxide dimutase, in presence of hydrogen, what happens? You are getting hydrogen peroxide. Now hydrogen peroxide will react with ferrous iron, which is present abundantly in blood, to form hydroxyl ion free radical. So these hydroxyl ion free radicals, the superoxide, hydrogen peroxide, hydroxyl are free radicals. They go and hit the DNA molecule and it damages. Along the side, these free radicals also damage the cardiovascular system present in our body. So it is cardiotoxicity is produced because of free radicals. Now, so this is how it binds. You can see visual effects, how this is the drug. You can see it here. See how it is binding and all this, the sugar molecules. So this is the drug DNA complex cross-linking. Now, in next slide, we will discuss little more here. You see, the activity of these drugs has been increased with the help of uh, formaldehyde releasing drugs or substance. We can tell they are aiding this activity. That is with the help of quaternium 15 and AN9, which is nothing pivalyl oxymethylene butyrate. These two, what they do is that you can see. So doxorubicin, along with this, any of these two, you can combine during the administration. What happened? There is a formaldehyde group extra forming. forming. Now what happens because of this? The cross-linking of DNA is faster. If you compare doxorubicin alone, instead of combining with this formaldehyde releasing substance, cross-linking is slow. Now, if you combine doxorubicin with formaldehyde releasing drugs, what happens? There is a cyclic formaldehyde conjugate is formed on the sugar molecule between amine group and hydroxyl group, and cross-linking is very fast. Cross-linking is very fast. Even with epirubicin, you can see it. Cross-linking is very fast. This is the advantage of formaldehyde releasing substance. Nowadays, they're combining with the doxorubicin and all this. Especially with doxorubicin, you can see it is very fast. Now, uses. Let us learn some of the uses of these anthracyclines all in at a time. Doxorubicin. Doxorubicin is used to treat solid tumors, benign tumors you can tell, as well as leukemias, lymphomas, and childhood tumors. Childhood tumors. Daunorubicin indicated to treat leukemia, acute leukemia. Epirubicin, breast cancer. Idorubicin, leukemia, hematological cancers. And it is given by oral route. Remaining drugs are given by IV route of administration. IV. And one more advantage you see, epirubicin and idorubicin are second generation drugs which have less cardiotoxic effects. Now one more drug we didn't discuss, we will discuss next coming class. Here it is used to treat mitoxantrone, leukemia, lymphomas, advanced breast cancer. So these are the uses. Now, one more important thing is that these drugs produce cardiotoxicity. That is what is the main reason? Because of free radical generation. One more reason is that sugar residue, which that also causes free radical generation, and also this drug interfere with rhinodone receptor, which plays very important role. Because of this, what happens is that you are getting cardiovascular disease, that is congestive heart failure is obtained. Well, not immediately or a period of time, depending upon the person to person. How can you treat this cardiotoxicity? By help of one drug, Dexrazoxane. This is the drug. You can remember like Michael Jackson. Dexrazoxane is antidote to be given for anthracycline. Then there is one more side effect, one more way to avoid cardiotoxicity is that you make the drug in the form of liposome. Then what happens? There is selective absorption by cancer cell, and there is no cardiovascular side effect. One more important side effect is extravasation injury. The drug eat the site of injection. You see, these are the pictures. The pictures you can see. These are how at the site of injection you can find redness, sometimes even blister if it is more toxic. Sometimes even you see the cell, it eats everything, and sometimes even danger. Even you can see it danger here. You see everything, the veins, tissue, everything is eaten by the drug. This is the issue with this doxorubicin. It is very good drug, more side effect again. To prevent this extravasation, this is called as extravasation injury. You can use that dexrazoxane. Along with that, cooling, local cooling should be done. Compression, cold compression should be done so that this injury is decreased. One more drug, Vinca alkaloids also produce extravasation injury, but there we use hot compression, not the cold compression. So when you give cold compression, what happens? Blood moves, blood circulation is improved, and drug also moves very well. Drug will not concentrate and in these places. So there is no injury. So this is how you can treat this extravasation. Now let us learn how this drug dexrazoxane will affect and prevent free radical generation. This is the drug dexrazoxane. Dexrazoxane. It is nothing but bis bis dioxo. There is two molecules if you can see. Dioxo, 2-dioxo, piperidine compound. Dexrazoxane is this belongs to bis, two molecules, bis dioxo piperazine molecule. This when you add into water, when it comes in contact with water or IV fluids, what happens? This is like amide group or lactam ring. The lactam is broken. The lactam ring is broken here. When it is broken, what happens? You find carboxylic group and amide group. That's why it is called as diamide diacid derivative of this dexrazoxane. Now, if you see the structure of diamide diacid is similar to that of EDTA. EDTA is a chelating agent. So similar to that of EDTA, this drug molecule, this active molecule, diamide diacid, will absorb the free radical, and those free radicals will not affect the cardiovascular system. So that is the advantage of this. That is the mechanism of this dexrazoxane. The structure, the active form of dexrazoxane is diamide, the diamide diacid, which is similar to that of EDTA. So that's why it is chelating agent. Chelating agent. So what happens is that it reacts with iron. So in the previous slide, we have seen iron is responsible for generation of free radicals again. Iron is generation. So this diamide diacid will react with iron, and when there is no iron, there is no generation of free radicals. There is no free radical generation. So this is how this drug acts. Now, this is how it acts. Then last drug, we are almost coming to end. Mitoxantrone. Mitoxantrone. This is the simplified analog. You see, this is the simplified analog. There is no A ring here. There is no sugar molecule also in this molecule. Only anthracycline derivative. What is that? BCD ring are present. No sugar molecule. That's why the cardiotoxicity effect of this drug is very, very less. And one more thing is that the four-membered ring is converted to tricyclic system. Tricyclic system. This system is responsible for intercalation. Intercalation. So this is also one more drug, mitoxantrone, which has less cardiovascular effects. From this, we can tell that the sugar molecule is responsible for cardiotoxicity. Cardiotoxicity. So let us learn in some questions. Now, please identify what is the drug structure given here. So what are the things, main important thing when you want to check? You need to check at 9th position what is the functional group present. and fourth position, and the sugar at the fourth position. These four you need to check. There is no hydroxyl group, so it is not a doxorubicin. It may be donorubicin. If there is donorubicin, it could have been methoxy group here. It could have been methoxy group. There is no methoxy group. That's why the answer is idorubicin. Idorubicin. Idorubicin is the answer for the given structure. Okay, so this is what we explaining. Now, which of the following heterocyclic ring is present in dexrazoxane? Dexrazoxane is a drug used as a chelating agent in treatment of doxorubicin, extravasation injury, and free radical generation. Free radical. It is a chelating agent. We told it has bis dihydroxy piperazine derivative. This is the functional group which is present. There is no phenoxazone. There is no anthracene structure. There is no aziridine structure. No, this phenoxazone is present in vilcamycin like that, and anthracene, glycoside, doxorubicin. Aziridine is mitomycin C. So this is the uh, this is the drug. What is that? Bis dihydroxy piperidine derivative. So you can see this is the molecule. This is the heterocyclic ring. Then one more last question, I guess. So which of the following drugs does not contain sugar molecule? Doxorubicin, methoxantrone, daunorubicin, all the above. In all these drugs, there is sugar molecule except methoxantrone. Methoxantrone, you can see the structure. There is no sugar molecule. Only B, C, D ring are present. Now, which of the following is the side effect of anthracyclines? See, cardiotoxicity, hepatotoxicity, abdominal discomfort, extravasation. So these two, A and D, are the side effects: cardiotoxicity and extravasation injury. So these are the references. So Foye, William Foye, Wilson and Giswold, Graham Patrick. Yeah, thank you very much. If you have any doubt, you can comment. Please subscribe to my channel. Bye.